Bailey, and today I'm going to talk to you about examining reinforcement speciation macaques using genomic data. So first of all, reinforcement is when hybridization between populations is selected against, resulting in speciation. So I have an animation to illustrate this where we might have populations of red and blue monkeys that border each other. Because of this, every once in a while they hybridize and form these gray offspring in the middle here. But if these gray offspring have reduced fitness compared to the parental populations, then we expect that hybridization would be selected against, and that would reduce the amount of hybrid crosses until eventually these populations cross only within themselves, solidifying them as distinct species. A common signature of reinforcement is reproductive character displacement, where reproductive traits differ between sympatric populations, those that overlap with each other, but not allopatric populations, or those that are geographically separate. And the reason for this basically is that geographically separate populations cannot hybridize in the wild, so we wouldn't expect selection against hybridization, whereas sympatric populations could potentially. And this phenomenon has typically been examined using morphological traits, but a review paper from 2018 details a methodology to examine this using genomic data. So I present a figure at the top here from that paper where basically they show that if we have some measure of divergence such as FST or DXY, we would expect the genome-wide average to be, for the most part, about the same between sympatric populations of different species and allopatric populations of different species. But in loci involved in reinforcement, we would expect increased divergence only between sympatric populations. So I plan to examine this pattern in the rhesus and cynomolgus macaques. So if you look at the map I have here, I have a picture of a rhesus at the top and a picture of a cyno at the bottom, and these sort of overlap their red and yellow ranges. So some notable distinctions between these species are that rhesus macaques are generally larger and more aggressive. They also have red sexual coloring on their face and down to their thighs, whereas cynomacaques only have a red sexual coloring above their thighs and not on their face at all. And also a notable trait is that cynomacaques have much longer tails. So their tails are about the size of their body length or larger, whereas rhesus have more, we would say, sort of medium length tails amongst macaques. And also in the contact zone, you can see in Indochina, there are clear genetic and morphological signatures of hybridization between these species. And so morphologically, that sort of shows as basically mixing of these traits to some degree. Some other facts potentially relevant to reinforcement are that introgression is predominantly from rhesus populations into cyno populations. As I mentioned, rhesus are larger and more aggressive, so that suggests that they could actually outcompete cyno males for cyno female mates. In addition to that, it's been shown in a laboratory setting that male cynos from Indochina are more aggressive than Indonesian cynos, and that suggests the possibility that these cynos near the contact zone have to be more aggressive to compete with introgressing rhesus individuals. And then on top of that, rhesus macaques sexually select for red facial coloring and darker coat coloring. And as I mentioned, red facial coloring is a trait specific to rhesus macaques and not cynos, so that suggests a sort of mating for one's own species. So my hypothesis basically is that if reinforcement speciation occurred between the rhesus cynomolgus macaques, there will be genomic signatures of divergence and divergent selection. I sort of detail in this figure here where I expect increased divergence between the parapatric rhesus and cynomolgus macaques, but no increase of divergence selection between the allopatric rhesus and cynomolgus macaques. So the first thing I've done here is that I obtained roughly 20 publicly available genomes for both these species from the NCBI. These were Illumina sequence genomes, so I ran them through a standard GATK best practices pipeline, where first I had FASTQ files that I mapped to a reference to generate BAM files. I marked duplicates and recalibrated base quality scores on these. And then I called SNP variants using haplotype caller, and I consolidated these and called genotypes with GATK as well. And lastly, I used variant quality score recalibration to filter these samples using, as a reference, known variants from the macaque genotype and phenotype resource. So then after that, I conducted my population genetic analyses with these samples. If you're looking at the images of the monkeys from left to right, I had 5 samples of rhesus from India, 4 Chinese rhesus, 6 Indo-Chinese Sinos, and 5 Mauritian Sinos. So these are ones from an introduced population originally sourced from Sumatra. So these are still sourced from an allopatric population. And essentially, I conducted genome-wide DXY measures between the parapatric populations and the allopatric populations, and I was particularly interested in regions that had a high difference between parapatric and allopatric DXY. With that in mind, though, I want to make sure that I define my windows and significance in as least of an arbitrary way as possible. So I implemented an R package called GenWin that I've used a figure from the associated paper here that has 4 panels, and I think illustrates how this works pretty effectively. Basically, if you look at panel A, you have some measure of divergence, some statistic like FST or DXY, and you want to calculate the statistic for all SNPs individually. Then in panel B, you can calculate a smoothing spline over these points. And then in panel C, you can identify inflection points from the spline, basically the dotted lines there. And then panel D, from those inflection points, you can identify new start and stop positions for variable sized windows and recalculate your statistics over that. So I went through that process with my DXY measures. And then to determine significance, I implemented another aspect of the GenWin package where it calculates this W statistic for these windows. So the equation for that is given to the right, and it's very similar to a T statistic. And one notable thing I'll point out about it is that you can see the variable N at the bottom right there basically refers to the number of sites contained in a window. So this statistic explicitly normalizes for different sized windows by accounting for the number of sites per window. That way you can compare windows of different lengths. So with that in mind, I extracted windows of the top 1% of W statistics and intersected them with the rhesus genome using bed tools. And I obtained outliers from that where basically we as a team, we input resulting genes into PantherGo, examined categories of interest such as locomotion, reproduction, behavior, coloration, interspecies interactions. That's shown at the pie chart at the bottom there. But additionally, we found that 8 of the genes that came from the 8 of these outlier genes were present in a list of 40 candidate aggression genes from a paper that compiled data from human GWAS, rodent transcriptomics, and causal evidence such as knockout mice and OMIM. And therefore, there's actually a significant overrepresentation of aggression genes in our data set as compared to the total number of aggression genes annotated in the rhesus genome. So I wanted to single out just 2 particular genes I thought were particularly interesting for time's sake. These are ATRN, which is involved in coloration. So it's been shown that in mice where ATRN is knocked out, they lose expression of a yellow band associated with the wild agouti type coloring, and that results in a mutant phenotype that's sort of dark or blackish. And another gene that counts is RBFOX1 that was part of the aggression candidate gene list. But in a variety of studies has been shown to be associated with aggression in various taxa, including humans, dogs, mice, and flies. And sequence variation in humans is associated with a variety of neural disorders. So with that in mind, for some future directions, I'd first of all like to basically obtain more samples. One thing I didn't mention is that there are plenty of publicly available rhesus genome samples, plenty of Mauritian Sino genome samples, but there's actually pretty lacking in publicly available samples for Indo-Chinese Sinos. And that's sort of the limiting factor in my study now. So in collaboration with Dr. Anthony Tosi at Kent State University, we plan to obtain more genome samples, sequence these from blood samples already obtained. In addition to that, I could calculate statistics such as FST and Tajima's D to reinforce my findings from DXY. And afterwards, I'd like to conduct further functional studies to find non-synonymous mutations in the outlier genes and sort of identify possible function of those. And as I show with all these logos here, there's no shortage of programs to do that with. So with that all in mind, I'd like to acknowledge various people in my lab and outside of it that have assisted with this project. And with that, I will take questions.